On behalf of the LAM Foundation, it is my pleasure to welcome you to this evening's LAMposium in your living room. My name is Cindy Beasley. I am the Director of Education and Advocacy for the LAM Foundation, and I will be your moderator this evening. In September of this year, over 400 LAM community members gathered in Kansas City for our International LAM Research Conference and LAMposium Patient Conference. This unique event brought together patients, family members, and friends, researchers and clinicians to educate, connect, and innovate LAM treatment and research. We begin this evening with highlights from the International Research Conference and will continue throughout 2026 to bring key LAMposium educational sessions to you. Thank you for joining us. Now, before we begin, please note that microphones will be muted and the chat box disabled throughout the presentation. At the conclusion of the presentation, we will open the chat box for your questions. There will not be our usual informal patient and family group discussion at the end, and we've done this so that we can allow for ample Q&A time tonight. So it is my pleasure now to introduce to you our distinguished speakers. Dr. Elizabeth Henske is the Director of the Center for LAM Research and Clinical Care and a physician at Brigham and Women's Hospital and Dana-Farber Cancer Institute. She is also Professor of Medicine at Harvard Medical School and Associate Member of the Broad Institute of MIT and Harvard. Dr. Henske's clinical and research interests include LAM, tuberous sclerosis complex, Birt-Hogg-Dubé syndrome, and angiomyolipomas. Her lab made the pivotal discovery that the sporadic form of LAM is caused by somatic TSC2 gene mutations. She also was the first to discover that the immune microenvironment regulates the proliferation of TSC2 deficient LAM cells. Dr. Henske served as co-chair for our International LAM Research Conference. Dr. Simon Johnson is Professor of Respiratory Medicine at the University of Nottingham, Director of the Nottingham NIHR Respiratory Biomedical Research Center, and runs the National Clinical Services for LAM and Rare Cystic Lung Diseases. Dr. Johnson's research group works on mechanisms and interventions for tissue damage in chronic lung diseases, including LAM, and on methods to improve diagnostics and management of rare lung diseases. He served as co-chair for our International LAM Research Conference. Now, if you ever doubt how dedicated and wonderful our LAM clinicians and researchers are, I would like to point out that Dr. Johnson is joining us from his home in England, where it is midnight. So thank you both for being here. I will now turn this over to you.
Thank you so much, Cindy. Can everybody hear me okay?
We can.
I am super happy to be here. I'm actually not in my living room. I'm in my dining room, and I'm in Cambridge, Massachusetts, which is just 3 miles outside of Boston. So Simon and I are going to go back and forth with the our slide. So Cora, could I have the next slide, please? So the first thing I want to do related to this conference and just LAM research in general is thank the truly amazing, stunning, wonderful team that represents the LAM Foundation staff. They are absolutely the best. Also want to thank Simon. Simon and I were on many calls planning this meeting, and I've known Simon for a long time, and it was just such an honor to work with him. And also our wonderful co-organizers for this conference, Vera Krimskaya, who is the Chief Scientific Officer for the LAM Foundation, Kayla Varberg, who is actually based in Kansas City and helped us with the idea of holding this meeting in Kansas City, which was really wonderful. And Harry Philippakis, who represents our next generation of LAM scientists and who helped with the poster session for this meeting. Could I have the next slide, please? So just to show you a little bit about what it looked like if you weren't there, you can see on the left one of the scientific sessions, 2 different shots, the front of the room and the back of the room. A really amazing thing was that the scientific sessions sold out. I don't know that that's ever happened before. And it reflects the, the intensity of interest in understanding LAM and figuring out how we can do a better job of treating LAM. The poster sessions you see on the top on the right, these are a very, very special part of scientific meetings where individuals, often younger scientists, can show data in a poster format, and then over the period of an hour or an hour and a half, everyone wanders around and talks to various people about their often very earliest research results. And you can see here Bill Stanford, who's a very prominent LAM scientist from Ottawa, Canada, looking at a poster during the poster session. And then we were lucky enough to have quite a few prior winners of the LAM Foundation Scientific Advancement Award present at this conference. That includes Frank McCormick, Nishant Gupta, Vera Krumskaya, Lisa Young, Joel Moss, and Simon and me, and also Jane Yu, who's standing next to me in this picture, who was this year's winner of the award. So next slide. So I think a highlight for me, maybe the highlight for me, was a talk by Frank McCormick, who many of you know. He was the first scientific director of the LAM Foundation. So he said something that really stuck with me, which was, what LAM has is hard-won, precious, and delicate. It needs to be cultivated and nurtured and never taken for granted. And I'll just read to you the 9 items that he identified that are our assets, things we're so lucky to have: a highly motivated, well-informed patient community, a global network of expert centers for clinical care and research, LAM clinical guidelines from multiple medical societies, highly effective patient advocacy organizations, peer-reviewed program for pilot funding for research, a clinically useful diagnostic biomarker, that's VEGF-D, a skilled, engaged, purpose-driven research community, that's the many people who were there in Kansas City, a solid understanding of the natural history and molecular basis of LAM, and then perhaps most importantly, an effective suppressive therapy, which is rapamycin or sirolimus. Okay, so Cora, could we have the next slide? Oh, sorry. Frank identified 4 really great goals for the future, and you'll see these goals reflected in many of the scientific sort of synopses that we'll be talking to you about today. So those 4 goals are to identify patients earlier, to develop methods to quantify LAM cell burden, to refine our current approaches to therapy and management, and to find new treatments that induce remissions. Okay, so now I'm going to hand it over to Simon.
Thanks, Lisa. We're going to kick off now reviewing the scientific presentations, and really this encompassed the sort of whole gamut of angles at which to tackle LAM, from improving the diagnosis, so identifying markers that can show that the person in front of them has LAM, understanding how the lung gets injured. And there's been a big shift, I think, over the years that both Lisa and I and a lot of other people on this call have been going to LAMposium, where there's now an increasing focus on how the lung gets injured, and mechanisms to prevent that specifically, rather than the previous focus, which was very much on mTOR signaling. And I think this is a positive thing, and there's a good convergence towards looking at the lung in detail now, and trying to prevent lung injury. And in line with that, we heard about several novel potential therapies for LAM and tuberous sclerosis. And additionally, other clinical research answering important questions for women with LAM. And I think anybody was there, I mean, it was a really fun and vibrant meeting, and really a lot of that is testimony to the staff at the LAM Foundation, who made it very easy for Lisa and I to help coordinate the meeting, and it made it a really successful event, I think. So if we can move on to the next slide, please. So the first area that we're going to discuss is the immune system in LAM, and you heard up at the start of the broadcast, Cindy highlighting that Lisa was pretty much the first person to highlight the important role of the immune system in LAM. And this is now one of the most promising areas of LAM research, and we had several cutting-edge presentations on this. Now, one of the first ones by Caroline Poole at Northwestern University in Chicago. And Caroline's been in the LAM field for a very long time. And her focus really is on immunotherapy for LAM, so boosting the response of the person's own immune system to target the LAM cells and destroy them, basically. And this, Caroline's presented, I think, at most LAMposiums for many years now. And on this occasion, she was describing a method that her team were developing to get part of the immune system called T cells. And T cells are immune cells that have different roles within the immune system. Sometimes that's killing cells that are either infected or foreign, like tumor cells, and you can sort of fit LAM into the sort of tumor cell bracket. Or they also help other immune systems to react to things. And then there are other subsets of T cells that balance the immune system and stop it becoming overactive. And so this is a complex system here. And what Dr. LaPoule is working on at the moment, and what she presented at LAMposium, was engineering specific T cells that react to a protein called PMEL, which is expressed by LAM cells. This protein isn't in normal lung tissue, but the LAM cell expresses a variety of proteins, which are usually seen in skin cells or the tumor melanoma. And Caroline works on melanoma and has adapted this therapy for LAM. So she's modified these cells to attack cells that express this protein PMEL. And she specifically targets this to the lungs by making the T cells express a receptor called CCR2, or chemokine receptor 2, which in many diseases targets immune cells to the lungs. So not only does she prime the immune cell to fight the LAM cells, but she tries to get them to go in the right place to do their work. And she showed studies that showed that this approach worked in animals. If we can move on to the next click, please. The second of the immune system presentations was from Katharina Meisel, who's based in Maryland. And she had two novel approaches in her work. She was using adjuvant therapies to boost the immune system. So your own immune system, again, targets the LAM cells and tries to kill them. And an adjuvant is a system or a substance which perks up the immune system. It makes it more active. And she used something called CpG, which is called cytosine phosphoguanine. And actually what this molecule does is it looks like the bacteria, it looks like the DNA of bacteria and viruses, and it sort of wakes up the immune system, particularly these T cells that kill things, so the cytotoxic T cells. And it reduces this other population of T cells that suppress the immune response. And so it changes the balance of how the body reacts to the LAM cells and makes it more aggressive. And this also, she showed, improved the response to rapamycin in her model systems. The other novel aspect that she was investigating was delivering these therapies into the lamb lung through the lymphatic system. As you know, the lymphatic system, the lymph part of lymphangioliomyomatosis is abnormal. And lamb nodules have little lymphatic capillaries in them, and the lymphatics in the lung are abnormal and related to the disease. So she has worked on a system where these adjuvant therapies are targeted particularly to the lymphatic to lamb by using the abnormal lymphatic system, and by engineering the size of these particles and their shape and their surface properties, you can make them travel through the lymphatics to exactly the place you want. So Dr. LaPoule targets the lungs by putting in this CCR2 receptor, Dr. Mysel targeting LAM by hijacking the lymphatic system to do that. So there's a lot of promising work now boosting the immune system, and I think if you go on to the next slide, I think that's Lisa describing some of the other approaches.
Thanks, Simon. So yeah, you've already heard a little bit about T cells, and I love doing the New York Times crossword puzzle, and this was, I think, maybe a year ago or so. T cell showed up in the puzzle. So on the left, you can see a normal T cell. It has all these what are called granules in it. And the granules contain enzymes that can help to kill other cells. For example, they can help to kill a LAM cell and eliminate a LAM cell. And what you see on the right is how lymphocytes often, T cells often appear in tumors. And we now know this is how they appear in LAM. They don't have that same killing ability any longer. And these are in oncology, these are called exhausted T cells, meaning that they just can't do that job anymore of killing other cells. So we'd really like to figure out why are the T cells in LAM exhausted, and how can we reactivate them? So the next slide includes some of quite a few different talks that were centering around why T cells and other immune cells are not active and what we can do about that. So the first that I'll comment on is from Yan Tang, who is my colleague at Brigham and Women's Hospital and Harvard Medical School. So she's been looking at how the TSC2 deficient cells, these are what we use as a model for LAM cells, might be influencing the immune system. And she found that a particular protein in the nucleus called NR2F1 is associated with a concept of dormancy. This means in tumor cells, or in this case, LAM cells, that are kind of in hibernation and can emerge at any time and begin to grow again. So she's found a particular gene called MDK, that stands for midkine, that is associated with this dormancy or stemness property, that sort of hibernation property. And she found that if she inhibits this protein midkine MDK, she can increase the ability of the T cells to be active again. So those exhausted T cells become more active. And when those T cells are active, they can start to recognize and remove the LAM cells. And she also found that this MDK focused treatment could also work synergistically with rapamycin. I guess that's a general concept that we saw in many different presentations. When people have a new idea that might be relevant to the treatment of LAM, many people are trying to understand whether this would work well in combination with rapamycin. And in this case, it seems that it would. If you can click to the next one, please. So then I talked about another way that we think the T cells are exhausted or not active. And that involves a protein called B7 B7H3. So B7H3 is found on the surface of LAM cells, and you can see at the bottom, I tried to draw that. B7H3 is the blue dots, and the sort of pinkish cells are the LAM cells. And what we found is that this protein B7H3 on the surface of the LAM cells has a powerful effect on the T cells. It actually makes the T cells exhausted. And as I showed you in the New York Times crossword puzzle example, you can tell that T cell is exhausted because it doesn't have those little granules that are what it needs to sort of attack and kill another cell. So if we get rid of B7H3 on the surface of the LAM cells, suddenly the T cells are active, and those active T cells can engulf and remove a LAM cell, which is what you're seeing in the cartoon on the lower right. So how can we do that? How can we get rid of the B7H3 on the surface of the LAM cells? Well, one idea is to use an antibody that would very specifically bind to B7H3 and cause the B7H3 to be stripped away from the surface of the LAM cells. And to test this, we've made some antibodies. We actually used an AI algorithm to make those antibodies, but then we wanted to test them. And so we used a relatively new kind of mouse called a humanized mouse. And Simon found this really cute picture of a mouse reading the newspaper to represent a humanized mouse. So humanized mice are given human blood stem cells. Usually these stem cells are obtained from umbilical cords, actually. So the human stem cells grow into blood cells, including T cells in the mice, and then we can test treatments that might be relevant to LAM in a human-like immune system. I guess that's another theme of this meeting, that we're recognizing that to really make progress in some areas of LAM biology, we've got to do it in a mouse that has immune cells like T cells. And in fact, much of the research that's been done previously in LAM has been done in mice that lack an immune system. So we're switching over basically in the field to doing many experiments in mice that have an immune system. They could either have a human immune system, like what we've been talking about here, or they could have a mouse immune system. So the next slide. So there's another kind of immune cell that we think is very important, and that's called a macrophage. Macrophage is Greek for large eater. And this talk was given by Joël Chamy, who works in my lab at the Brigham and Women's Hospital. So we know that there are two kinds of macrophages. There are many kinds, but we kind of lump them into two groups. So the one on the right is that's sort of yellowish looking, is representing what we call an M1 macrophage, or a pro-inflammatory macrophage. And these are macrophages that respond to an infection in the body, for example. And they also can recognize and sort of eat up a foreign cell or a bacterium or a tumor cell or a LAM cell. I kind of think of macrophages as like a cellular Roomba. They're scanning around all over the place, picking up stuff that doesn't belong there. But then there's another kind of macrophage that's depicted in purple. And these are called tumor-associated macrophages, or M2 macrophages. And this is like a Roomba where the battery has run out. And if you ever had a Roomba, it's when the battery is getting low, it's supposed to go back to its home base and plug itself in and recharge. But this is like a Roomba that got stuck under the couch or something and can't get recharged. So it turns out that in many kinds of cancer, we see these exhausted or Roombas with no battery, these M2-like macrophages. And we also see those macrophages in LAM and in angiomyolipomas. In fact, in angiomyolipomas, macrophages are one of the biggest cell types. And they're almost all these macrophages that are no longer able to recognize and destroy foreign objects. So what Joelle found, working with Tina Liu, who was a member of my lab and now has her own lab in China, was that if we convert the M2-like macrophages, the purple ones, into the yellow macrophages, and we could do that with a drug that was made by an investigator at the National Cancer Institute in Bethesda, just a few buildings away from where some of you may have met with Joel Moss. So this drug, if we give it to mice, converts the macrophages from the purple M2 one to the yellow M1 Rumba-like macrophage, and it dramatically shrinks tumor size. Makes the LAM cells much smaller, makes the LAM cells go away. So we think in the future, we might want to be thinking about targeting or treating LAM by making the T cells more effective and simultaneously making the macrophages more effective. And that those two things together could allow the body's own immune system to recognize the LAM cells, realize that they don't belong there, and just get rid of them. And this is, of course, you may know this is how we treat many kinds of cancer right now, with drugs that restore the immune system's ability to recognize tumor cells. Okay, so I'm gonna, if you could do the next slide, please, and I'm gonna turn it back to Simon.
Thanks, Lisa. So we're going to change tack a little bit now and look more specifically at lung structure. And these next couple of abstracts are under the theme of what's called spatial biology. So when we used to go to LAMposium in the old days, we'd look at the expression of one gene or maybe one protein in a cell or a group of cells. Now the technology's moved on, and this allows us to look at all the genes in a tissue. And so you can not only see which genes are expressed, and you can do this with proteins as well, which proteins are expressed, but you can see where they're expressed relative to each other. And that means you can then work out by the signature of all the genes and the proteins which cell you're looking at, what type of cell you're looking at, what that cell's doing, and which cells nearby it's talking to. And this is called spatial biology, and it's sort of revolutionizing a lot of what we do. One of the leaders in this field, and particularly for LAM, is Yan Zhu in Cincinnati. Yan described a new study, a very big piece of work that she's only able to describe a small part of, about defining how LAM cells and the cells that talk to LAM cells in the lungs are arranged. And she introduced this concept of a LAM niche, which is the part of the lung where the LAM cells sit and they interact with other cells. And so she described two sorts of LAM cells. They're both LAM cells, but they just express different proteins or different genes, and they do slightly different jobs within the LAM nodule. We've known for quite a long time that some LAM cells grow faster than others from work from a long time back that Dr. Moss did at the NIH. But now we're able to understand this in a lot more detail. Yan was also able to describe different types of LAM-associated fibroblasts. We've known about these for 10 years or so, and they're not LAM cells, but they're attracted to LAM cells and they support them, prevent them from injury from the immune system and other things, and they contribute to lung damage. And so these sorts of data tell us various things. They tell us a lot more detail about how the LAM cell interacts with other cell types, how it causes injury, and it's starting to tell us where LAM cells come from. One of the things that's come out of this work over the last few years is that the genes expressed in LAM cells look quite similar to genes expressed in cells in the uterus. There are, and a lot of people think maybe the LAM cells come from the uterus because of this. There are other possibilities. The talk wasn't really about this, but this spatial biology now is able to give insights into these types of processes. And the picture here, the little different colored dots here, are the different types of cell within a LAM nodule, and the little diagram underneath with the different sized blobs are representing how strongly genes are expressed in these different cell types. And so you can see that some of the LAM cells will produce extracellular matrix proteins. Some of them will produce chemokines that attract other cells. And this tells us how these cells interact. And by understanding these processes, hopefully we'll be able to tackle them better in the future. Another representative of the Cincinnati group, who I think is a graduate student, Grace Wickenheiser, used another spatial transcriptomic method, so looking at gene expression across a tissue to understand how the different cells within these nodules were interacting with each other. And there were other people that used similar techniques in their presentation. And this is another really promising area that a lot of people are now using to understand LAM. So if we can move the slides on, go on to a little bit about slightly more biology focused abstracts. And so quite a lot of people were working on different aspects of signaling and proteins and lipids that are produced by LAM cells, and how they contribute to disease progression and how you can target these. So Jane Yu at the University of Cincinnati has worked on lipids in LAM for quite a long time, and she presented work this time about sphingosine. Sphingosine is a lipid, and it's a building block for larger lipids. And she looked specifically at an enzyme called ASAH1, which is the name for its acid ceramidase. Acid ceramidase breaks down big lipids into sphingosine and fatty acids. And so this enzyme, if you have a lot of it, you have a lot of sphingosine. And sphingosine not only builds lipids, but also can have biological effects on cells to make them grow and survive better. And on the whole, sphingosine is bad for LAM, Jane has shown. This ASH1 acid ceramidase enzyme, people have known for a while that it's elevated in cancer, and it's also elevated in LAM. And so what happens is ASH1 is increased, so there's more sphingosine, and Jane showed there was more sphingosine 1-phosphate in LAM, and also the receptor, that the protein on the cells that reacts to the sphingosine 1-phosphate is also increased in LAM. So you get more of the protein and more of that makes sphingosine, and then you get more of the receptor that causes the biological effect. And so blocking this enzyme, Jane showed reduced SP1 signaling and the growth of these TSC2 null tumor cells in mice, suggesting that maybe targeting sphingosine metabolism and this enzyme specifically that produces more of it might in the future be a helpful intervention for LAM. So move forward, please. So yeah, Jane Yu used to work in Lisa's lab, and then the next person here, guess what? She used to work in Lisa's lab also. Lisa's responsible for all these high-level LAM researchers around the US. So this is Magdalena Carbonacek, who's now at Texas Tech, and she was studying something called extracellular vesicles. Extracellular vesicles are tiny packages that bud off cells, and inside them they have proteins and other material that signal to other cells nearby. And what Magda showed in a really elegant and complex piece of work is that LAM cells produce extracellular vesicles at quite high levels, and that these vesicles can talk to other cells and encourage these cells by interacting with proteins on their cell surface called integrins to invade into tissues around them and spread around the body. And one of the things that we know that LAM cells do is they can spread through the body and seed in other places and cause new LAM nodules to form. And Magdalena was looking at one of these the mechanisms of this mechanism. Right, move on again, please. So I don't think Dr. Diaz has been in your lab, Lisa. Maybe I'm wrong, but this was the third sort of LAM biology abstract, and Dr. Diaz was looking at this this protein N-myristoyl transferase 1, or NMT. And this is a protein that is involved in the function of lysosomes. Lysosomes are small small subcellular particles that are responsible for the turnover of proteins. And importantly for LAM, the mTOR complex, the signaling pathway that's strongly activated in LAM, relies on being on the surface of the lysosome for part of its activity. And Dr. Diaz has shown that inhibiting this NMT protein that supports lysosomal function will reduce the efficiency of or reduce mTOR signaling in both cancer and LAM cells, and inhibitors of this protein can cause LAM-like tumors to shrink in mice when they're treated with this drug. So all of these abstracts are sort of looking at different ways to reduce the growth and activity of LAM cells, and all really quite different approaches to this problem, which is encouraging. Right, so next slide, please. So here we're going to move on to talking about fibroblasts and extracellular matrix. So as I said earlier, we know that LAM cells in the lungs attract fibroblasts to them. Fibroblasts are cells that everyone has. Their normal cells. They don't have TSC2 mutations. And their normal function is to generate extracellular matrix, amongst other things. Extracellular matrix is a sort of scaffold. It's a protein scaffold that all cells sit on when they grow. And apart from acting as a scaffold, this extracellular matrix can program the cells to behave in certain ways, depending on which proteins are present in the matrix. We didn't know until relatively recently that in the lungs of women with LAM, particularly in the LAM nodules, there's actually quite a lot of extracellular matrix. So the little figure that's up here is a picture of a LAM nodule, and this is stained by something called picrocereus red. And so this reddy pink color is actually staining extracellular matrix in a LAM nodule. So you can actually see in this LAM nodule that quite a big percentage of it is this extracellular matrix protein. Normally, you don't have this much matrix in this sort of place. This is an abnormal finding, and what we found is that this extracellular matrix is deposited from LAM fibroblasts into LAM nodules, And when you take a LAM cell, or the model LAM cell, the 621 cell, and you grow them on this this LAM-derived extracellular matrix, they don't respond to rapamycin as well as if they're growing on ordinary tissue culture plastic in the lab. So this extracellular matrix, we think, might be blunting the effect of rapamycin. We were able to show that when cells grow on this LAM matrix, lots of genes get increased in cells. Some of these drive the cell through the cell cycle, so they make it grow, and this happens even when rapamycin is present. And we discussed that there are drugs that target some of these genes, particularly one called samuraciclib, which targets a protein called cyclin-dependent kinase 7, which drives the cell cycle. So we think maybe targeting these extracellular matrix-sensitive pathways along with rapamycin might be slightly better for treating LAM, particularly in patients with more advanced disease who have more extracellular matrix in their lungs. So yeah, if we can move on. The second collagen fibroblast extracellular matrix type paper came out of Vera Kremskaya's lab. And Vera took a slightly different approach from our lab. She started by categorizing using single cell sequencing, one of these methods where you look at all of the genes that cells express, and identified in her case, 3 different subpopulations of LAM cells. And one of these LAM cell types strongly produced extracellular matrix, these scaffold proteins that I was talking about. And Vera postulated that this extracellular matrix that gets produced perhaps shelters or hides the LAM cell from the immune system, makes it less accessible. Actually, if you look at the little LAM nodule and how much matrix there is in there, you can see that maybe that's possibly the case. Different populations of these LAM cells in her study had different sensitivities to rapamycin, different roles in lung injury. And Vera was able to show how these LAM cells were activated, and how you could block the production of this extracellular matrix in LAM cells by using mTOR inhibitors. So she showed that if you use rapamycin, collagen secretion didn't go down very much at all. Whereas if you use one of these newer mTOR type inhibitors, so-called bisteric inhibitors, which bind to more than one part of the mTOR complex, you get a more efficient suppression of collagen, suggesting perhaps that different types of mTOR inhibitors may have different effects and possibly better effects than rapamycin. Next slide, please. So the final piece of the LAM fibroblast matrix abstracts came from Emily Liu, who works in the Ryan lab in Iowa, and she looked at a protein called TGF-beta. It's been known for a long time that TGF-beta activates fibroblasts, the cells that produce extracellular matrix, and it causes them to secrete collagen and other extracellular matrix proteins. Emily showed that TGF-beta was expressed quite strongly in the lungs of patients with LAM, and it was activated in LAM lungs. So this protein that activates fibroblasts and makes matrix, she showed was upregulated. So all of these 3 abstracts looked at similar sort of area from slightly different angles, all adding to the strength of these findings. The other thing that they postulated was that this TGF-beta also perhaps reduced the efficiency of repair in the lung, which is a whole other area and another potential area to target. Okay, so if we move on to the next sequence of abstracts, which is about modeling LAM. Now, models are important because when we see something or we have an idea about how LAM might work, we usually try and design a model system to see if that works. So it has some components of the whole disease, not all of them, but usually it's set up to be able to test a particular hypothesis, a particular idea to see if that's true. So you saw a picture of Bill Stanford earlier, looking at the posters in Lisa's introduction. So Bill's a stem cell biologist in Ottawa, Canada, and he uses stem cells, and stem cells are cells which can differentiate or turn into other cell types. So stem cells sit in different places in the body, waiting till there's an injury or an organ to be formed or modified, and then they differentiate to make the cells that are required. So they're sort of an undifferentiated cell type that sits there waiting to repair things by turning into the cell that's needed. And in research now, you can program these stem cells to turn into whatever cell that you like, really. And Bill's been using stem cells to model LAM and angiomyolipoma-derived cells for some time. He started with a neural crest type cell, and a neural crest is an area of the embryo in development which sends cells into different parts of the body to form different organs. The reason that Bill was interested in the neural crest is that LAM cells have a strange mixture of melanoma-related antigens, the things that people use immunotherapy against, and smooth muscle antigens. And one of the cell types that you see that combination in a neural crest, and for quite a long time, and I think probably still to some degree, people thought LAM cells perhaps were derived from the neural crest. So Bill's used these cells to make model LAM cells. He does this by knocking out the TSC2 gene, the gene that's faulty in LAM. And these cells develop a gene signature that looks like standard LAM cells when you measure them in people. Two interesting things. One, he found that male and female cells have different gene signatures when you do this to them, which is interesting considering the strong female preponderance of LAM cells. And he also identified a new relationship between Schwann cells, which are cells which support nerve cells, as a possible relation to the LAM cell. And I think that's a bit of a watch this space thing. And the point of all this work is to develop a system that you can screen drugs and screen knockout genes to identify therapies that can work alongside rapamycin to treat LAM. The other abstract in the modeling section is from Amy Ryan. Her student described the TGF beta project that you saw in the last slide. Dr. Ryan, who's a principal investigator in the lab, is studying how these fibroblasts that are important in LAM interact with the lymphatic endothelium. And the lymphatic endothelium is a part of the LAM nodule. And really, not many people are studying this at the moment. So this is a sort of welcome development to understand the contribution of this. And she really described relatively early stage models. And one of the innovations that she brought to her work is she developed models that breathe. They don't really breathe, but if you think about when you breathe in and out, your lungs are expanding and contracting, and your cells are all experiencing that stretch. 12, 15 times a minute, they get stretched and they contract again. And when you do that to cells, that changes the genes that they express and the proteins that they make. And it's an important part of their environment. So adding this factor into modeling it's potentially important to discover new areas that you otherwise might miss. Yeah, if you move on to the next slide now, I think Lisa's going to cover these abstracts on LAM diagnostics.
Thank you, Simon. Wow, I'm so enjoying listening to you explain all of this, Simon. It's great. Okay, so three important talks in this area of LAM diagnostics. The first was from Elias Bu-Farhat. He works in David Kwiatkowski's lab at Brigham and Women's Hospital in Boston. And many of you may know that David Kwiatkowski is really the world leader in the genetics of LAM and tuberous sclerosis and many other things. So we already knew that at least some cases of what we call the sporadic form of LAM, that means LAM that occurs in a woman who does not have the disease tuberous sclerosis. We've known for some time that if you look at LAM cells, they have TSC2 gene mutations, leading us to conclude that LAM in the sporadic form is caused by these TSC2 mutations. But it hadn't been done very many times because it's very technically challenging. So we didn't know how often it's TSC2 and if there could actually be any other genes that might be able to also cause LAM. And this is a really important question because we want to know for sure the cause of LAM so that when we're modeling it, for example, we're modeling the right thing. So David and Elias looked at the DNA from 70 different women with LAM in the lung tissue. And they looked for mutations in about 12, 15 different genes, including TSC1 and TSC2, and other genes in the pathway that were plausible candidates to possibly cause LAM. And they did this using a very advanced form of mutation detection that allows Elias and David to find even to identify TSC2 mutations, even if it's only, say, 5% of the cells in that piece of lung that carry that mutation. So the bottom line is pretty amazing. They found TSC2 mutations in 62 out of 70 of these specimens. And they did not find mutations in any other gene. So I was looking at this as a sort of glass half full that, wow, almost all of the LAM, sporadic LAM has a TSC2 mutation. But during one of the question and answer sessions at LAMposium, someone said, wait a minute, I thought it was always caused by TSC gene mutations. What's going on here? Why isn't it 100%? Could there be another gene? So we don't know the answer to that. My hunch is that no mutation detection, especially for a large gene like TSC2, is going to catch every single mutation. So TSC2 has 41 little pieces, what are called exons. And the mutations could be in any one of those 41 pieces. They could also be somewhere between those pieces between the exons, preventing the gene from being correctly assembled. So I'm guessing that even, the best TSC LAM geneticist in the world using the best possible technique is still not going to have the ability to detect every single mutation. But based on these data, I think we have to leave the door open that perhaps there could be another gene somewhere that causes LAM. But the most important conclusion is that most cases of LAM are associated with TSC2 gene mutations. The next really interesting and important talk from this section was by Minzi Guo, who's at the University of Cincinnati and works closely with that team there that we've heard mention of many other wonderful scientists who are in Cincinnati. So Minzi is an expert in the bioinformatics of large data sets and using those large data sets to make discoveries. So in this case, Minzy is looking for proteins that might be present in the blood that could be used to improve the diagnosis of LAM, or perhaps to be more sort of tuned in to whether a particular individual with LAM is likely to have that LAM progress or not. So he's doing that by starting with the single cell data. That means all the RNA molecules from each and every cell in the lung of someone who has LAM. Simon mentioned some similar work earlier. And looking for which of those genes might make a protein that would leave a cell and be found in the blood. And then he's using the spatial transcriptomics that was also mentioned by Simon, where you can look at all the cells in LAM relative to where they are next to their neighbors, for example. And using this kind of discovery method, potentially find proteins in the blood that we could use to better diagnose or treat LAM. And then the last talk in this section was from Taigan Han, also goes by Adam. So he's in my lab. And this work is actually kind of the counterpart to what Minzy is doing. So Minzy is using bioinformatics to identify proteins in the blood. And what Adam, and working closely with Remy Dessler, also from my lab, did was to use a really amazing technology called SomaScan. So SomaScan can measure 10,000 proteins in one drop of blood, less than a drop of blood. And it's really just amazing. It's not only measuring proteins that are supposed to be in the blood, like proteins that are known to circulate, like albumin or molecules that bind lipids or TGF-beta, which Simon mentioned earlier, but lots of other proteins that might just leak out into the blood at very small amounts, but can still be detected there. So what Adam in my lab found was that one of the proteins that leaks out into the blood in women who have LAM is actually PMEL, which was a protein that Simon mentioned several slides back to be a target that Caroline LaPoule has been using to identify immune strategies for LAM. So we found that PMEL was in the blood of women who have LAM, and it's not present in the blood of healthy control individuals. And it's an amazing thing because PMEL is the protein that a pathologist would look for if someone has a biopsy for LAM to make sure that it is in fact LAM. It's almost like the hallmark of LAM. So we found PMEL in the blood as well as other proteins, and we developed a score that in this very small group of women with LAM, only 20 women with LAM and 10 controls, but in this small group, could identify everyone who has LAM, including women who don't have a high VEGF-D. So this so-called biomarker score could be useful for the diagnosis of LAM, especially in women who have a low VEGF-D level and therefore might need a lung biopsy. And we're hoping that future iterations of this kind of a score could also allow us to understand the response to sirolimus. So could we develop a more precise measure of whether someone is responding to sirolimus or not, or what dose they need, for example. And actually, I should mention that this whole study, the SomaScan study, was done thanks to Joel Moss and his whole team at the NIH and many women who may be on this call. This is the study that Joel asked women who are just starting on sirolimus to travel to the NIH and take their very first dose of sirolimus right there at the NIH so that we could have carefully matched blood specimens drawn just before that first dose, just after that first dose, and then these women came back 3 months later to have their blood drawn again so that we could measure these proteins in the blood before and after they received sirolimus. And it's just one of many examples of how amazing this community is, that someone like Joel Moss and his team was able to do this very precise collection of samples. And of course, all these women had many other analyses like lung function testing. So on the one side, we have Joel Moss with his team, and then on the other side, we have the women with LAM who were willing to fly to or travel to the NIH to be part of this study, which we call the U01 study. So many, many thanks to the women with LAM, and many, many thanks to Joel Moss and his amazing team. Okay, the next slide. We're almost done. I think we have 2 more slides with data on them. All right, so what about imaging? You saw the comment from Frank McCormick at the beginning that we really need ways to measure LAM more precisely so that we could know if it's getting better or worse and not use lung function as our main measure. So Carmen Priola, who's also at Brigham and Women's Hospital, has been using a new kind of PET scan. And this technique is called metabolic imaging to try to detect angiomyolipomas and measure their activity very precisely. So you probably heard of a PET scan. We do them pretty often in cancer. The kind we do for cancer uses a tiny amount of radioactive glucose, and PET stands for positron emission tomography. So because cancer cells in general take up much more, they absorb much more glucose than surrounding normal cells. They need a lot of glucose. If you do this in someone who has cancer, the tumor will be very bright because it's taking up a lot of glucose. So Carmen is doing something similar, but she's using acetate, which is a building block for the formation of lipids. So she has been giving a tiny amount of radioactive acetate to humans with angiomyolipomas. She's doing this in collaboration with Massachusetts General Hospital. And you can see that the arrow is pointing to that red circle. That red circle is the uptake of acetate PET by an angiomyolipoma in a woman who has LAM and also has an angiomyolipoma. So super, super exciting early results. She's also seen that this can be suppressed by the use of rapamycin. And then the last imaging talk was from Bilal Ismail. He's in Cincinnati and works in their MRI scanning group. And he was using a newer type of MRI called xenon MRI. And he actually was doing this in women who were participants in the MILD trial, MILED. This is a trial that we should know the results of pretty soon, where I think about maybe about 60 women with LAM who had early LAM, but had high likelihood of the disease progressing, were randomly assigned to either a low dose of sirolimus for 1 year, 1 or 2 years rather, 1 milligram of sirolimus, or a placebo control. And the goal of the study is to see whether this low dose of sirolimus can prevent LAM from progressing. So the xenon MRI was done in a subset of individuals from this trial. And the results seemed very encouraging, that this xenon MRI could detect changes in the lungs that correlated well with lung function, like FEV1. There should be one more data slide. Oh, yes. Okay, so I'll give it back to Simon.
Thanks, Lisa. Yeah, so this is one of the clinical studies that was done at LAMposium, and it was coordinated by Henske and Dr. And they were trying to build evidence to see whether sirolimus is safe during pregnancy. And so the the problem here is, as most of you will no doubt know, is that pregnancy in LAM has been associated with the high rates of pneumothorax, and in some patients, accelerated loss of lung function. And so for some people, especially those with lower lung function at the start, pregnancies is a risk for patients. And as sirolimus stabilizes LAM, the question really is, could it stabilize LAM during pregnancy particularly? The difficulty is that there's very little data on human pregnancy in people taking sirolimus. There's a few case reports from patients who take it for kidney transplant, but there wasn't a lot of data for patients with LAM. And animal studies have suggested that sirolimus can reduce the growth of animal fetuses if taken during pregnancy. So we really don't know whether it's safe for people. And so along with LAM clinics in the USA, us in the UK, Brazil, France, China, the combined might of all these LAM clinics was only able to find 12 pregnancies for people who'd taken sirolimus whilst they were pregnant. The 12 pregnancies resulted in 10 live births, and those children are now between 1 and 6.5 years old. The little graph here says mysterious scribbles appeared on the slide. But the little graph here is lung function from a selection of the patients where it was available during before their pregnancy and afterwards. And you can see, although these lines go up and down a little bit, they're essentially very steady. And this is not normally the case in pregnancy for people who aren't taking sirolimus. The other thing is that, as I said, pneumothorax is very common during pregnancy. In this study, no one whilst they were taking sirolimus when they were pregnant had a pneumothorax. And so it looked like the sirolimus was effective, a very small number of people. This is 12 pregnancies in 10 people, I think. The sirolimus was very effective at stabilizing their disease during the course of pregnancy. The other good news is that the children, although most of them are still infants, didn't seem to have suffered any adverse consequences by being exposed to rapamycin when their mothers were pregnant. So this was a useful study to try and understand what advice to give to people with LAM who wish to become pregnant about whether to continue with rapamycin or not. I don't think we have any hard and fast answers, but this very small study looks relatively promising, I think is all I'm really prepared to say on that. But it's an important question for women with LAM. So I think that concludes the data. And I think Lisa's just going to wrap up now.
So I just want to end with such a strong thank you to the LAM Foundation team. This was front and center in the strengths that Frank McCormick highlighted. We have a highly effective patient advocacy organization, but maybe the most effective patient advocacy organization I've ever encountered. They not only made this conference run incredibly smoothly, but just every single day they bring their best selves to help us move things forward on every front. And then if you just click one more time, I'm going to end with the goals that Frank outlined, that I hope you've seen. Many of the scientific presentations that Simon and I highlighted are designed to move one or more of these forward, to identify patients earlier with biomarkers, for example, to develop methods to quantify the LAM cell burden, for example, the imaging, the xenon imaging or the PET imaging, to refine our current approaches to therapy and management, and to find new remission-inducing treatments. So we're going to end there, and I think maybe we'll turn it back to Cindy. Thank you guys so much for being here tonight.
My gosh, this was amazing. Thank you, Dr. Henske and Dr. Johnson. Really, you mentioned the staff a lot, but I am always in awe of all that you do for our community and your dedication. And I know, Dr. Henske, I knew you back in the day when I was first diagnosed, which was over 25 years ago. So you're still here with us, still fighting for us. And I hope you can see all of these applauds and hearts and feel the love that we have for you and Dr. Johnson and all of our researchers and scientists. We are so very, very grateful. This was such an informative and important presentation. You made very complex ideas very palatable for us and understandable. So thank you so much. That is a gift as well to translate that to a lay audience. We're very appreciative. I'm going to go ahead and ask Erin if you will open up the chat box, and we'll see if we have any questions there. Or if you can remove the slides. There we go. And I need to turn my chat box on here. Okay, so we have, sorry, technology. Here we go. In the various studies that have and are being conducted, have any uncovered possible treatments to improve lung function and breathing for LAM patients?
I would say that we're making a lot of progress, but there is nothing right now that I believe would be better than what we have, which is sirolimus or rapamycin. And that's a good thing because we have a really great treatment right now, and it's going to be hard to find something that's better. So we're all working on that, but we don't have anything right now that we think would be better than what we have, which is a good problem to have. We've got a really good drug right now.
Mm-hmm. Great, thank you. And I want to, the one thing I've learned being on staff is there are only 5% of rare diseases that have a treatment. And the fact that we have one and we got one so early in the process is nothing short of miraculous. And again, thanks to you all. All right, so we have another. Is there a potential for a gene therapy treatment? Was this discussed at all at the conference? I know it's a big buzzword in the science community. So
there wasn't anything on gene therapy as such. There are people talking about supplementing TSC2 in the lung using various technologies, but I'm unaware that any of these have moved into actual studies. Lisa, I know that TSC2 replacement strategies have been mooted by some people, by some small companies who specialize in this type of research. I'm unaware of anything that's been moved to patients yet.
That's correct. We're actually just starting to work on that in mice a bit. Just got a grant to do that, but we don't know anything yet.
Mm-hmm. Great, thank you. And I know when we hear information like this, we as LAM patients get really excited, and we want that bisteric inhibitor tomorrow, right? So someone is asking, what is the timeline for these studies? And I know it's all individualized. Is there a way that people can learn more about these studies, maybe to understand a little bit more about timeline and what that might look like for us as we get excited?
Yeah, I know the timeline is such a hard thing. So we first would have to be convinced that something was really better in the models that we have. That could be the stem cell model that you heard about from Canada, or it could be one of our mouse models. And then we would have to prove that this new treatment was at least as good as, or ideally better than, rapamycin. And that's why these biomarkers are important, be like the imaging biomarker or the drop of blood protein biomarker. right now it's pretty hard for us to know whether something is better than rapamycin. Again, it's a good problem to have. Rapamycin works really well. So I think we're in a little bit of a holding pattern right now. It'd be hard for us to prove that something was better than rapamycin. I don't know what you think, Simon, about that.
Yeah, I mean, having an effective treatment makes bringing new things in actually more difficult. As Lisa says, you've got to show it's going to be better than that. Or even if you use it alongside it, because the change in the disease parameters when someone's on rapamycin has been leveled out quite significantly. So you're looking at small changes in somebody who's on rapamycin. It's quite hard to show that something can do that in a clinical trial setting unless the effect's really strong. And so 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 evaluating these things is difficult, and I think one of the questions there is about timelines. And many of these drugs that we've been talking about, things that affect the immune system, for example, are already in clinical use for other diseases, so you can bring those to patients more quickly than a brand new drug where you have to test the safety extensively. So I mean, I think there's there's hope that there'll be things not too far away, but it's not an easy thing to get over the line, really, to show that it really is beneficial.
Thank you. I have a specific question here. Have any of the studies looked at disrupting the extracellular matrix, hiding the LAM cells from the immune system?
So it wasn't presented at the meeting, but there's a drug for pulmonary fibrosis, which is basically an extreme form of extracellular matrix deposition in the lung, which is another horrible lung disease with quite a bad outcome. And there's a drug called nintedanib that stops the formation of extracellular matrix in that condition. And the drug nintedanib that they use for pulmonary fibrosis has been recently tested in patients with LAM in a study in Europe run by Sergio Harari, who's in Milan, in Italy. And there were some beneficial effects. The effects were small, but I think there were some beneficial effects on lung function, which was quite stable when these patients were taking nintedanib in this study. So that was a direct use of an antifibrotic drug in women with LAM. Nintedanib also blocks kinase receptors, which are expressed on LAM cells, and the effect may have been due to an alternative effect on the LAM cell. We don't really know the answer to that, but it looked like there may have been some benefit, and it's certainly known as an antifibrotic drug. So yeah, I'm not sure what Dr. Harari's next plans are for nintedanib and LAM. Possibly a bigger study, I guess.
Thank you. And then, when we start diving into the world of research, we all have various understandings of how funding works, what lab does what. One of the questions here, and maybe you can give us a broader picture, particularly Dr. Henske, on it, says, what studies are dependent exclusively on NIH funding? So in what you presented today, were any of those, I don't even know how to approach that question, but because I don't understand that part of that side of things either. So can you help us understand
First
that a
of
little
all,
bit?
thank you for asking. So NIH is the National Institutes of Health. It's where Dr. Moss works, and it's also the source of funding for most individuals in the US who are doing any kind of research. So I'm glad you asked, because some of you must know that the NIH budget has been very severely affected in the past few months, and we desperately need that funding to continue to do what all these things that are happening in LAM. I couldn't tell you precisely what amount of the research that we talked about tonight was supported by the NIH, but it's probably on the order of maybe 75% of it. So if you have an opportunity to advocate for federal support for research, this is where the money goes to help figure out the cause and treatment of diseases that affect all of us. And I think there's some amazing statistic, maybe you know it, Cindy, about rare diseases, that something like 1 out of 10 individuals in the United States has
Correct.
something that is officially categorized as a rare disease. And often, actually, when we study rare diseases, we learn things that help us understand many other diseases at the same time. So if you have an opportunity to stand up for research, please do so.
Yes, and I know Dr. Moss really, and we try to do this at the LAM Foundation as well, is really encourage folks to be a part of his protocol. One of the strengths of that protocol and one of the reasons why it has not been touched so far by cuts is because of the LAM Foundation community coming and being a part of that for so many years. So another great opportunity, kind of two sides of the coin, advocating for more federal funding for research, which we have a whole page dedicated to that on our website, but then also participating in those NIH studies with Dr. Moss is also very important. So I'm going to probably close with this one. I will tell you all, I see many questions in here, some that kind of are a little bit outside of the scope of what we're talking about. I've got one I'm a little confused on, so we will reach out to you individually to help get you these answers. And if we need to touch base with either Dr. Henske or Dr. Johnson on something on these questions, we will. But I'm going to ask this one because I do remember back in the day talk about using statins to help with LAM and trying to understand what are other things that we could combine. So one of the questions here is, are you recommending impairment of the mTOR pathway at other points than rapamycin, for example, metformin or statins? You know, what type of research is out there, or what is your thinking on that?
Maybe I could start by just saying that I don't think there's anything that we know of that's better than rapamycin right now. And I don't think that we have any data that combining rapamycin with a statin or rapamycin with metformin would be better than rapamycin alone. So I wouldn't suggest that anyone take that approach. Of course, if you need to be on metformin or you need to be on a statin, there's no harm in being on rapamycin in those, but I wouldn't do it because of hoped for benefit for LAM.
Great. And is that being looked at? Is this, when you guys approach different types of research, you know, somebody even brought up this Dr. Evercure, you know, Dr. Feigenbaum, who had started this organization with Castleman's disease. He discovered rapa was a therapy for his disease. So what is that role when we start hearing things about GLP-1 or statins or other things like that? How should we be thinking about that in the LAM community, and how do researchers think about that?
I know, Simon, do you want to answer that one?
Yeah, I mean, in terms of the position we're in at the moment is that we have a larger number of potential drugs that may be helpful for LAM based on all of the things we presented this evening and years of previous research, and that moving these things towards seeing whether they're effective treatments for patients is really the hard part, and what they call the translational gap, translating these laboratory studies to improve the lives of people living with LAM. And that's a tough thing. I think some of these things have somewhat fallen by the wayside. There was a lot of excitement about statins for a while, as you said, Cindy, I don't know, quite a few years back now, and a couple of slightly contradictory reports about whether they're helpful or not. And there was a small trial which didn't show a lot of benefit, but probably wasn't big enough to do that. And I think this is the difficult position that we're in at the moment in the field about bringing these new findings forward to see whether they actually benefit patients rather than just models. In the UK, we have a new rare respiratory disease network. Lisa's on the advisory board of it, actually. which is really aimed at addressing the translational gap, using new strategies, including giving very small doses of drugs, too small to be toxic to humans, to see what the effects in actual real people are rather than animals, to look at novel ways of doing clinical trials in rare diseases, and I hope that we can address some of these issues.
Great, thank you so much. Well, I'm going to start wrapping us up here. We are almost at the 8:30 mark. So Dr. Henske, Dr. Johnson, thank you again for being with us this evening, for all that you do and have done to move LAM science forward. We are forever grateful. And as I talked even earlier, you all are the most incredible people I know on the planet. So I wish you both a good night's rest. For you, Dr. Johnson, with the hours that remain, and Dr. Henske, thank you for taking time out of your evening to be with us, and I hope that you enjoy the rest of your evening as well. So as we close, I want to remind you all of the importance of tissue donation in furthering LAM research. And somebody had a question in there about some brain tissue. If you are a LAM patient living in the US and are having an upcoming surgery of any kind, please reach out to the LAM Foundation for information on how you can donate tissue through our partnership with the NDRI. We can certainly explore those things. It is a simple process with exponential impact. I don't know if you noticed tonight, several times Dr. Johnson or Dr. Henske would talk about, we looked at the LAM tissue and this is what we found. And that's because of you, and that's because of this community who've been dedicated to donating tissue. So very simple, very important thing that we can do as LAM patients to move research forward. Our next LAMposium in your living room will be Wednesday, January 21st, where Susan Jacobs from Stanford Medicine will be presenting on the topic, Understanding Supplemental Oxygen, Device Options, and Tips for Staying Active. So we will have several of these LAMposium in your living rooms throughout this year that will cover actual sessions that were covered at the in-person LAMposium conference. So I do hope you will join us and watch your emails for that. Recording of this week's session will be available on our website next week. So thank you all again for joining us. Have a wonderful rest of your evening.